I'm going to do something that is a little unusual, which is read some text, because I tend to introduce people kind of ad lib. And the reason for that is you'll see in a minute. But the first one is because, this is a special lecture that we have once a year. It's a Tryon lecture, and I need to give a little bit of a historical background for the lecture. And so I'm just going to read the blurb that we've been passing along year after year for this lecture, Eric, so that you know exactly where the history behind this. So this lecture is named after Robert Tryon for his scientific contribution to psychological inquiry, for his loyalty to University of California, and because of his deep devotion to the Department of Psychology and his colleagues who form its faculty. The friends of Professor Tryon chose to commemorate his research career by establishing an annual lecture in his name. The chosen lecturers are to be scholars of distinction addressing biological aspects of psychology, Tryon's own era of inquiry. Tryon was born in Butte, Montana on September 4th, 1901. He received his AB degree at Cal in 1924, and then his PhD here in 1928. He continued at Berkeley as professor and chair of the psychology department. He died in Berkeley, California, September 1967. In pursuit of understanding the biological contributions to the behavior, Tryon used iterative breeding and testing of rats to perform experiments to support a genetic basis of behavioral differences, laying groundwork for the field of behavior genetics. He also made pioneer contributions to cluster analysis methods. In studies of human subjects, he developed theories on the social ecology of human behavior. Tryon is remembered as a professor devoted in service to this university and its students. He was involved in campus life through diverse groups, from the graduate council to the faculty committee on athletics. During his tenure as chair of the Department of Psychology, he introduced a faculty committee system for sharing in the governments of the department, at that time, a radical change. It has been said that Tryon was a tolerant man and willing to look carefully at any scientific proposition seriously advanced, that his response to intellectual challenge was typically one of enthusiasm, and his verve and delight served to stimulate the creative powers of those around him. It is in the spirit of creativity and innovation, in thinking about the biological basis of behavior, that I am delighted to announce our 2022 Tryon lecture will be delivered by Erich Jarvis. So sorry for reading that out, but Eric, that gives you a little bit the background for this lecture. And I hope you can tell, Eric, that there's no better person than you to do this, right? I mean, this is the person who started behavior genetics, and to those of you who don't know Eric, I mean, he's a giant in this field, right? He's introducing you is tough, just because of all the contributions that you've made at all these different levels. And so anyway, I'm reading that. I'm going to read a second one, just because it's another way of introducing our speaker before I'm going to go and do quickly, hopefully, more traditional introduction. So the second thing that I'm reading is a little paragraph in the Science kind of news and views about the life of Erich, and I think it's short, and it quickly kind of quickly and sharply defines him. And it says, as a child of mixed ancestry growing up 5 decades ago in the Harlem and Bronx neighborhoods of New York City, Erich Jarvis was immersed in the teaching of civil rights leaders. From Martin Luther King Jr., he took the precept to love everybody, accept everybody. Malcolm X inspired him to succeed by any means necessary. Both principles have served him well in science, says Jarvis, now a prominent neurobiologist. Jarvis is a natural collaborator and also an opportunist. I try to accept any kind of tool or any type of technology that I need to use to answer my question, he says. So for those of you who do not know Erich, I really highly recommend that you go to his website, you go to his CV, and you check it out, because it's so impressive. You're going to be blown away by all that he did himself as a person in terms of growth in the field of science, in terms of obviously the contributions that he made, and I'll mention that just very briefly, and in terms of his efforts for diversity and racial equity in science. It's just it's really an example. See, I mean, Eric, you're a star, and I know that you know it, but we also know it, and we appreciate it, and the feelings of awe, they're here, right? I'm really just very impressed. All of this, and I've known Eric's not very well, not personally, but in interactions in scientific meeting, because we overlap in birdsong, and he's always interested in what I'm doing. He always has questions. He's always friendly. It's just a pleasure to just know him personally and to be able to spend time with him. Okay, I haven't actually done any kind of like formal introduction, so let me try just a little bit of that as the last thing. And I don't want to take too much of your time, Eric, because I know you have very important things to say. So this is the kind of classic, kind of more boring intro that you would get when you introduce somebody. It tells them a little bit about his career in terms of the different dates where he achieved different things. So he's really a product of Rockefeller University. He got his PhD there in the laboratory of Fernando Nottebohm in 1995. He stayed as a postdoc for 3 more years. He became an assistant professor in the Department of Neurobiology at Duke University in 1998. By the way, the same year that I started a faculty position at Berkeley, which is very humbling when I compare my CV to Eric's CV. In 2008, he became a Howard Hughes investigator. And in 2016, he went back to Rockefeller University, where he is a full professor and the director of the Rockefeller University Field Research Center and the co-director of the Rockefeller Vertebrate Genome Laboratory. His contribution to science is impressive, both in its quality and its quantity. He has over 200 publications. Many of these are published in Nature and Science. I think, Eric, you should have a section in your CV that says Nature and Science publications. It just lists those, His work is really on the neurogenomics of complex traits, including human language. He's done incredible work in birdsong, looking at the genetic basis and the evolution of vocal learning in birds, not only in one species, but doing this comparative and evolutionary approach across multiple species, and has, by doing that work, established a connection between the animal kingdom, birds, and humans that is important for so many reasons, in terms of understanding where language has evolved, but also in I think making us closer to animals. I think I'm going to stop there. I could go on and on. I have a list now here of your different awards that I was going to say, but I need to stop. Please go and look at his CV, and it's a fascinating story on its own. So with all that, Eric, welcome. I'm so glad you are able to take time from your schedule to give us this lecture, and I'm looking forward to it.
Well, thank you for that introduction. And yeah, I know something, but I didn't think that as much as that, Gloria, as you mentioned. But it's always good to see you at conferences. Good to meet up with Linda as well, who we overlapped at Rockefeller, and good to be amongst the community here that I understand is coming from a diverse background of scientific interests and otherwise. So I hope to do Tryon, that is justice. So let me go ahead and share my screen and then the PowerPoint. Okay, can you see it? All right, and I have to share with the audio because I have video. All right, share sound. There we go. All right, can it be seen? Great. All right. And
Eric, we see all your entire desktop. I don't know if that's what you meant.
Yeah, something happened. Let's see. Desktop share. Oh, I think I have to undo my presentation. That's a little strange. Okay. It caused my PowerPoint to close. Now I'll bring it back open. Let's try this again. All right, do you see the PowerPoint? Great, okay. Well, now you guys see what's busy on my desktop as well. So, all right. Well, thank you, Linda, and thank you, Frederick and others for inviting me for this lecture. So today I'll talk about our work on the neurobiology of an evolution of vocal learning and spoken language and the relationship between the two. And I encourage you to put questions in the chat as we go along, which I'll pick up at towards the end. If there's a pressing question in the middle, don't hesitate to ask me, Linda. And I chose to study this trait actually even since my days as a graduate student because it's a complex trait, but not maybe so complex that it'll never be solved in terms of spoken language. It's something that's rarely found, as in these pictures up here. A few species have it and some of our close relatives don't have it. And I won't be able to get through everything today, but one of my favorite reviews I wrote back in 2019 on evolution of vocal learning and spoken language. I encourage you guys to go read that as well to find out more. Some things have changed since then, but a lot of it is still up to date. And so broken this down into three levels of analysis: the behavioral, the anatomical, and the genetic. And then we'll end off with testing some hypotheses with genes that we found. And so starting at the level of behavior, we humans often, I think, don't appreciate when we think of spoken language that it's a multi-trait ability, even though to us it might feel like it's all one wrapped in one big ball. And there are certain components to it that I like to think about, or Tecumseh Fitch, who also proposed this, that are specialized or to humans, like vocal production learning, the ability to imitate and produce sounds. But we don't realize there are other components like vocal usage learning, the ability to make associations with or do a command with sounds that you hear, or auditory learning is more ubiquitous amongst the animal kingdom, but they alone don't make spoken language. So I like to define it as a specialized form of learned vocal communication with some components found in most vertebrates and a highly specialized vocal learning component found in only a few species. And who are those few species? There's 5 groups of mammals and 3 groups of birds that have this ability of vocal production learning. Us humans, of course, and then dolphins and whales together called cetaceans, bats, elephants, and seals, and songbirds, hummingbirds, and parrots. And, but even some of our close relatives or our pet animals will have auditory learning and can understand some of the human speech sounds, but can't imitate. And so, for example, you can teach a dog to understand the word sit in English, or siéntese in Spanish, some rudimentary sentences like get the ball and so forth. But a dog can't say, okay, you got it, I'll sit, right? Instead, a dog goes woof. And it can learn how to woof in different contexts, what we call usage learning, but not actually imitate sit. Whereas this non-human species, a parrot, a budgerigar, which is basically called a parakeet because it's a small parrot, this animal's been raised with humans for 4 years and learned up to 400 words and can produce them in new combinations that he didn't actually learn. This is quite remarkable. Not all non-human vocal learners are this good, but this is a lot of English words that were imitated and putting them in new combinations and so forth. And some might ask, is disco and other animals who mimic other species, are they mimicking those sounds just to mimic, but without meaning and so forth? But you can see that disco, although a large percentage of the time, disco seems to be rambling off words randomly. But a lot of times disco and other animals who imitate human speech will actually produce them in meaningful context. And in this case, when this dog shows up, disco goes meow, kind of gets the category wrong. The owner did have a cat, by the way, but still gets the idea that this is another animal. Think about that.
Yeah, for sure.
And so that's the best you can get out of a non-human animal that's a vocal learner. And here is the best that's thought to get out of a non-vocal learner, Coco, a gorilla. And when this study was published, people got excited and reporters were calling me up and asked me, what do I think? And let me just play it for you.
How about when you're coughing? That was good. You did a sneeze and then a cough. Excellent.
So, okay, I just hit something. Let's see. Hi, can you see me still? Hold on a second. Show video panel. There we go. All right. So here, what's going on is that
how about when you're
Coco was raised with humans for 39 years, and with an effort to try to teach Coco how to imitate human speech sounds, a female gorilla here, but Coco could not learn how to produce any of these sounds. Yet, as you can see here, Coco has good auditory learning. Coco understood human speech sounds, not the full 20,000 vocabulary repertoire a normal human would, but several thousand words. In this case, how about coughing? And Coco understood the question and tried to make a coughing sound and did make a coughing sound and is able to make mostly voiceless kind of sounds, meaning it's not modulating the vocal tract. It's getting air through the vocal tract, but she is not actually modulating in a way that we humans do or songbirds do for the syrinx, for modulating that air to produce imitated sounds. And so this distinction has caused myself, many other scientists like Frederick and others, to really be asking the question, what is the difference going on here that someone closely related to us, like these non-human primates, can't even get out a simple word like apple, whereas those more distant related to us, like 300 million years distant, like these parrots, can go much further and say golden delicious, or all different kinds of varieties of apples. And so, the answer to that question of what makes the difference here is something that I've been trying to pursue for many years. And I'm going to argue it's in the anatomy, and there's more than just one difference. But my proposals haven't been the only ones around. There have been many for vocal learning and spoken language. And I put 6 popular ones here that I'll just quickly walk through. One difference, people argue, it's a difference in brain size, particularly the forebrain. So in green is the forebrain regions in a cartoon fashion here. Red is the brainstem motor neurons that control the vocal organs. And blue is the actual vocal organs themselves. And so some argue that vocal learning species, and particularly humans, have a bigger forebrain, bigger cortex, and that allows more brain space for the vocal, auditory, and other circuits to develop to give you spoken language and vocal learning. Some argue that it's not really a difference in the brain, it's a difference actually in the peripheral organ, that the larynx is descended more in humans, or it has more complex muscles in songbirds that allows them to produce more varieties of sounds from a similar brain. Some argue that it's not the peripheral musculature or a difference in size of the brain, but the presence or absence of particular circuits that control the vocal organ, the haves and the have-nots. And when you have the cortex controlling the vocal organs, then you can get complex vocal communication. Some argue that the circuits are there, but it's either makes a direct projection from the forebrain to the motor neurons for vocalizations versus an indirect one, or the direct projection is there, but it's enhanced in the vocal learning species. And whenever you have direct cortical control, it's thought you have more fine motor control of your behavior. Others argue it's not how the forebrain communicates with the outside world of the brain, but it's in the central nervous system itself, or in the forebrain itself. Like one hypothesis is that this arcuate fasciculus from secondary auditory areas to Broca's areas is either not present in the non-vocal learners or non-human primates, it's there in humans, or enhanced in humans. And finally, mostly coming from the linguistics community, some have argued that there's a separate language module in the brain that only humans, maybe vocal learners, have some higher level cognitive areas that then the auditory and the vocal and other circuits and the manual gesturing circuits feed into and get all its commands from this language module to tell these other circuits what to do. And what you'll see in this 2019 article, and since then, reviewing the literature, I argue that these three hypotheses I boxed in are the most supportive given the scientific evidence. That is, humans do have a larger brain relative to body size than other primates and other mammalian species. And the vocal learning birds actually have twice the density of neurons in the same space as the non-learning birds, and birds in general have greater density of neurons. So you get a parrot-sized brain is really the size of a small non-human primate. There is some evidence, we'll go through this, of presence and absence of circuits, or ones that have more greater connectivity versus weak connectivity. This language module idea, I just don't, in my view, find good support of it. This kind of connection to the auditory to frontal vocal motor regions we find in mice, I'll show you that later. And I think Tecumseh Fitch has debunked this hypothesis about the descent of the larynx. So focusing on a few of these, moving forward, the work that I've contributed to this area began as a postdoc here at Rockefeller, where we found that when songbirds sing, in this case, a canary singing its learned song, that act of singing of a learned vocalization is associated with rapid increases of immediate early genes in the very brain regions that control the production and the learning of the sounds, like HVC and area X here. And what you see in white here is a signal for a messenger RNA of CFOS, the EGR is another one of these genes. And in red is the crestoviolet staining. And this is the front of the brain, this is the back. You can see the cerebellum here, not showing activation, by the way. And what we found is that the more a bird sings in a half-hour period, the more activation of these genes that we see in these song learning brain regions, and a pretty robust response. For every one song about the bird sang, there was a one-fold increase in the area exon nucleus for this immediate early gene. And there's a whole field of studies now on what these immediate early genes are doing in the brain for learning and memory and so forth. But we use it also like a molecular mapping tool, like a functional MRI tool, where we found that this singing-driven gene expression was motor-driven. The bird had to sing, and even if you try to sing but don't get the sounds out, you will see this activation. And you don't need to hear, and you don't need to have somatosensory feedback. And so using it like an MRI tool, we map vocal communication circuits across various bird species, including the vocal learners, parrots, hummingbirds, and songbirds, and the non-learning ones like quails and pigeons. And what we found is that in all these bird species, in blue is color-coded the auditory pathway. They all had auditory systems activated when they hear species-specific sounds. But only in the vocal learners can we find these forebrain circuits in yellow and red here in songbirds that involved in the production of learned song and in the acquisition in red here in the vocal learning birds. And these forebrain circuits overtook this brainstem circuit in gray here that you find in all vertebrate species that vocalize in the midbrain and the hindbrain that control the vocal organs. And what you see is this novel forebrain system has taken over. And looking at this phylogenetic tree here on the left, we interpreted at that time, either there were 3 independent gains of vocal learning, and each time there were 7 such brain structures in each of the 3 vocal learning bird groups with similar connectivity. Or maybe there was a common ancestor back here somewhere, and there were 4 independent losses, that then can explain this result here. Or maybe everybody has it to various degrees and it's independently amplified in these species. Or something I didn't appreciate at the time as a neuroscientist, maybe the phylogeneticist got the tree wrong, right? And all these vocal learners, birds are closely related to each other, and that's why they have such a similar pathway. And there is actually a fourth idea that this picture here has ended up in some religious textbooks, saying that this has to be because of intelligent design, because how could you get such a similar circuit? And we didn't really have an answer. And before I get to in getting to that answer, one of the things I got involved in is sequencing genomes of many different bird species, not only to find genes associated with this vocal learning trait, I wanted to find out if the tree was right or wrong. And a number of other people studying birds wanted to have this answer as well. And this tree here on the right is based upon DNA hybridization. There are others based upon single genes. And when you have genes based upon single or even 10 genes, you can have lots of errors in the species relationships. Here now we have the first genome scale, entire genome that is alignable across all birds representing bird orders. And we found that the old tree was wrong. Lots of rearrangements in species relationships. And what about the vocal learners? Well, actually, the hummingbirds were pulled much further away from the songbirds and parrots than in this tree over here. And the parrots and songbirds are brought a little closer together. So it indicates at least 2, if not 3 independent gains. And to have a common origin for birds, you would have to go back to the time of the mass extinction of dinosaurs, and the surviving species that survived that mass extinction be a vocal learner and all these other lineages, 10 independent losses for penguins and pigeons and vultures and so forth, not to have the trait. So the most parsimonious explanation now is that it is independently evolved. What about humans? Well, if these vocal learning birds can come up with a similar solution over the last 65 million years, are we going to be any different? And so we began to ask that question. And the first lesson you learn here is a songbird brain, a zebrafinch. And here it is that same brain to scale with the human brain. You can fit roughly 3,000 zebrafinch brains, the size of the tip of my thumb, here into a human brain. And so brain size doesn't matter. Neither does cortical surface of folding matter. We argue, when I say we, the community in general argue, it's the presence or absence of a particular type of circuit that matters, that you can scale upwards or downwards. Brain size matters more in terms of body size than mostly anything else. And so what is that circuit? And so this is now showing sort of an update of what we originally published back in 2004. That circuit looks like to be similar in the vocal learning species. When you take the MRI lesion and other functional literature in humans and non-human primates, what you get is as follows, that we humans have a Wernicke's auditory and primary and secondary auditory cortical regions that I argue is homologous with what's found in non-human primates and found in all vertebrates that have auditory pathways going to the forebrain, including in birds. And just organized differently in terms of its layered and cellular organization. And this is why I think dogs can understand sit, siéntese, come here, get the ball, or Coco can understand how about coughing, because Coco has a Wernicke's-like area, maybe more advanced in humans, but it's not presence or absence. But we humans and these vocal learning birds seem to have evolved novel brain regions involved in the production of learned speech or learned song, and an acquisition of it, like Broca's area, and all things complex for speech and production. And having similar connectivity of cortical basic ganglion thalamic loops for this learning circuit, this production circuit, taking over this innate brainstem circuit that controls innate vocalizations in non-human mammals that then control the vocal organs. And so, in primates, there might be a ventral laryngeal cortex that have some rudimentary functions, but nothing like what you see in these more advanced circuits in humans with getting that auditory input. How could it have come about? This figure has also ended up in some religious textbooks as, intelligent design. And here is the alternative answer to that hypothesis, or let's say that idea. And that is, we found that not only the vocal learning brain regions have this vocalizing or motor-driven gene expression inside them when the birds sing, but the very surrounding regions, circuits, also have this motor-driven gene expression when the birds are performing movement behavior. In this case, the zebrafinch hopping in a rotating wheel for the first time in its life. Without sound input, it doesn't require hearing, it doesn't require somatosensory feedback, it doesn't require vision as well, because this guy is hopping in the dark. And we found this was the case for all vocal learning bird species. There were these movement-driven gene expression regions around the vocal learning regions in all vocal learning species. And in the non-learning species, we also found the movement-driven gene expression regions, but we did not find the vocal learning regions inside of them. That is, there was just movement region, non-vocal movement regions only. And when we look at the connectivity of the vocal learning circuit and the connectivity of the directly adjacent neurons, they have similar connectivity. Again, a cortical basal ganglion thalamic loop, which I'm going to argue probably controls motor learning how to move, but not vocalizations, and a descending motor system that projects out for other motor neurons for wing movements, arm movements, and so forth in mammals. And the circuitry is similar with some differences. For example, in the small nucleus RA, it makes a direct projection to the motor neurons that control the voice, whereas the surrounding motor neurons make indirect projections to what's called the reticular formation, then to the motor neurons that move the other body parts. And whenever you get direct projections, as I said earlier, you get more direct fine motor control from the forebrain. And it has a collateral synapse here for the surrounding areas that goes to the striatum area here that you don't see in the vocal learning circuit, but in mammals, you do see this collateral for other circuits outside the vocal systems. And so, so then we asked, well, how could this have come about? And is something similar going on in humans? And the answer came around the same time from a colleague of mine, Steve Brown, who is studying dancers, and found that when he, in PET imaging systems, when he gets dancers to do learned choreographed movements, they show greater activation, the highest activation in areas that are directly adjacent to, in this case, laryngeal motor cortex regions, ventral laryngeal motor cortex, and Broca's area, and also parts of the striatum involved in speech production. And so this led to what we are calling the motor theory of vocal learning origin, where I've argued that all species that vocalize, which is most vertebrates, not, minus some reptiles and fish, all species that vocalize have these brainstem circuits that are involved in the production of innate vocalizations. They all have motor learning circuits that control learning how to fly, learning how to walk, learning how to do the circus tricks or that your dogs can learn how to do for a circus or elephants and so forth. And that consists of two of these pathways. And I argue that during embryonic development, these motor learning circuits is replicating multiple times to control different muscle groups of the body. And that in the vocal learners, like songbirds and humans, I argued that this pathway, through some kind of gene mutation, replicated one more time. You got a whole brain pathway duplication that took over the brainstem motor learning circuit for vocalization, brainstem innate circuit for vocalizations, to get an emergent vocal learning pathway that's similar to the surrounding circuits. And this is how you can get something that's similar in humans and songbirds and parrots, and not something by intelligent design. And so if this is true, then the genes that function inside the vocal learning circuit should be similar to the ones that function outside of them. And the connectivity also should be similar, as I mentioned before. And the development of these circuits should be similar. And so this is what we went to test next. The genetic underpinnings of this motor learning circuit, what we think it could be, and the vocal learning circuit. And the way we went about that is to laser dissect out the vocal learning brain regions on thin sections of the bird brain underneath the microscope. So we had like you see in a CSI movie, we're dissecting the cells off the glass slide, putting in a little tiny Eppendorf tube, laboratory test tube, isolate the RNA and proteins as well. And we do the same thing to the surrounding brain regions. And we do this for all the vocal learning birds and non-vocal learning bird species, pretending if they had a brain region for vocal learning, we dissect that same region out as well. And we profile them at that time to what was called oligomicroarrays, where they're glass slides that have little spots of nucleotide sequences for thousands of genes that are regulated in the body. And the Allen Brain Institute for Brain Science was doing something similar for human and non-human brains, where they had 3,700 samples from 6 people and 252 samples from 6 monkeys, including motor cortex regions, profiled them to microarrays, and students and postdocs in my lab at the time, now who have their own research groups, developed the algorithms and also generated the bird data to compare the gene expression profiles inside versus outside of these soil learning nuclei to look for gene expression specializations shared with humans or maybe not shared. And what we found is that out of 7,000 genes that we can find expressed in the brains of songbirds and humans, there were 75 of them, 75 of them, that showed convergent expression, up or down regulation in the songbird area X and an anterior part of the human's basal ganglia that was later found to be active during speech production, particularly vocal learning in humans. And 55 genes were found to be convergently up or down regulated, and the songbird RA that makes that direct projection in human laryngeal motor cortex also thought to make a direct projection. And so out of all the brain regions it could match throughout the entire cortex, basal ganglia, cerebellum, spinal cord, and so forth, this region had the most similarity to the human laryngeal motor cortex, more so than it did to a non-human primate brain cortical regions or a non-vocal learning species like quails. So here, songbirds and humans are more similar to each other in this specific brain region for these specific set of genes. And here's what the expression profiles of some of those genes look like. Each row is a gene. Each column is a sample from a particular species. Blue is downregulation compared to the surrounding motor cortex. Red is upregulation. And for this laryngeal motor cortex region, we have lots of downregulated genes here in the 3 vocal learning bird groups and in the human speech motor cortex here. You can see the pattern is more similar than in the motor areas of the non-vocal learning birds like dove and quail, or other areas that are not speech cortex in humans or even non-human primates. Many of these genes are involved in axon guidance functions or neural protection functions, and we'll get to why they might be downregulated later. We tested one of them by in situ hybridization, or several of them, in this case SLIT, one an axon guidance molecule, which makes sense since the connectivity of these regions differ from the surrounding motor circuits, and we demonstrate its downregulation in the vocal learning brain region of all of these bird species, but not of quails and doves. In humans, as well as parrots, by the way, we find 2 regions of differential gene regulation in the primary motor cortex, which we call the dorsal and the ventral laryngeal motor cortex. And around that same time, Eddie Chang's group at UCSF was putting electrodes into patients where they're removing tumors in epileptic regions to try to preserve the speech region, and found 2 regions, not just one laryngeal motor cortex, but 2 that are precisely the coordinates we find in humans with the specialized gene regulation that have the highest activation of neural firing when we humans are producing speech. And so that showed a functional parallel there. So we were all excited with these 100-something plus genes shared between humans and songbirds, and now we wanted to study their function. And so around this time, we started to try to reclone these genes and get them into expression vectors or gene manipulation vectors. And we started to find that these genome assemblies that we're using at the time were highly fragmented. So here's what we call a alignment of the SLIT1 gene, and each one of these little tick marks here in black are gaps in the genes, and they were in protein coding sequences, so we couldn't get the complete gene. And what we're finding, we're missing parts of genes, or entire genes were missing or incorrectly assembled, and it was causing biologically meaningful repetitive regions we're finding in vocal learning species in regulatory regions of these genes that were being misassembled because it's hard to put repetitive regions of the puzzle together. This is causing many months for students and postdocs in my lab to clone and correct the genes. And many of us were unknowingly working with artifactual gene structure and sequence and making false conclusions about the biology. And so the leaders of some of these big genome initiatives, what was called the Genome 10K Consortium at the time, to sequence 10,000 vertebrate species, came to me and said, Eric, what you did with those 48 bird species and your vocal learning studies, could you please help us do this with the broader vertebrate genomics project in general. And I kind of, I reluctantly agreed because I'm a neurobiologist, but I was the neurobiologist who brought all the genome people together. And so I agreed to do this, and we formed what we call the Vertebrate Genomes Project with this ambitious goal of one day within the next maybe 10 to 20 years, sequencing the genomes of all 70,000 vertebrate species on the planet. So I can get all my vocal learners done and the non-learner close relatives and everything else. And we're in the first phase now, representing all vertebrate orders. And what we made our mission is not just generating all this genome data, but at high quality. So it's error-free, near gapless, it's chromosomal level, phased paternal and paternal chromosomes, the haplotypes, make sure all genes are annotated correctly, and then make sure we use these genomes for specific question at each of these phases to address fundamental questions in biology, disease, and conservation. And our first set of publications on the first 16 species generated with high quality long reads that filled in a lot of these gaps that the old assemblies had. We published in a special issue in Nature, and the editors decided to call it the spinal frontier for sequencing all these vertebrates. And I made sure that one of the species we focused on most to get as high quality genome assembly as possible was a hummingbird, a vocal learning species. And so having this more complete genome now, and now using new technology, not only oligoarrays with glass slides with lots of genes spotted, but RNA-seq, which is profiling the expression of every single gene inside of a cell. Now, instead of a few genes we see specialized in these vocal learning circuits, in songbirds, we see 25% of the genome has specialized expression in one or more song learning nuclei. That's quite remarkable, right? Of your 20,000 genes, 25% of them is specialized up in red or down regulation in blue. And what's interesting is that most of these song learning nuclei have specializations specific to them, like here in LMAN, or here in HVC, or I'm sorry, yes, in LMAN or HVC or area X. LMAN has the most specializations compared to all the song nuclei. So that was the first lesson. And here is just showing a Venn diagram that most of them have like 2,300 genes specialized to LMAN alone and not HVC and RA. And in human, still looking at the microarray data, so it's not RNA-seq yet, we're working on that, but a similar story like laryngeal motor cortex, supplementary motor area Broca's have more specialization special to them than shared specializations. And so now taking these higher quality genomes in this greater set of specializations and a more, let's say, finer view of avian brain organization color-coded here, we found that validated with these additional genes, or found additional genes, that area X is convergent with the anterior part of the human striatum. So validated that, validated the RA result, particularly to dorsal laryngeal motor cortex, but also further found that HVC was also convergent with human dorsal laryngeal motor cortex for 69 genes, if we just look at dorsal cortex alone, or 250 if we combine it with other regions, the ventral laryngeal motor cortex, that is. And so, and when we look at this heat map of the genes convergent between songbirds and human speech regions, we find that the majority of them are specific to either HVC or RA, but not both, as before. Here is, genes that are shared, but this is indicative of maybe HVC and RA matching different cell types in human laryngeal motor cortex for its conversion. Fortunately for us, the Allen Institute generated another recent data set where they profiled different cell sites from layers 2, 5, 6, and so on from the human primary motor cortex. And we looked at the region that the bird RA was in general, the archopallium, in which HVC was part of the nidopallium. And we find that these cell types in HVC and the surrounding nidopallium has an expression profile of layers 2 and 3, supporting a hypothesis that Harvey Carton proposed many years ago. Whereas RA had an expression profile that matched human laryngeal motor cortex layer 5 neurons that specifically project out of the telencephalon and down to the brainstem and the spinal cord, like RA does itself. Michael Brainard's group with Todd Roberts' group also generated single cell sequencing of HPC and RA itself as well. And we took their data and asked in their data, where are the convergent genes with humans showing up? And they're precisely showing up in the cell type that projects from HPC to RA. So layer 2 and 3 intratelencephalic projections supporting that conclusion with humans, and the cell types in RA that project out of the brain, the forebrain, and make that direct projection. So not just any neurons, not the inhibitory neurons or other excitatory neurons, but those specific cell types are convergent. And when we look at the functional gene categories in those cell types that are convergent, what we find remarkably here is for the RA and HPC specialized genes matching human laryngeal motor cortex, they're specialized for genes that are downregulated in for neural system development, axon guidance, neural projection, and so forth. Not upregulated, but downregulated are the enriched categories. In contrast, in area X, the genes that are convergent with the anterior striatum of humans are upregulated for myelination, glial cell differentiation, oligodendrocytes, and cell membrane functions for this learning nucleus. And so this prompted us that maybe there might, ask the question, how are you doing this to all these different genes? And once again, when we published some of these data earlier on, certain religious groups said, how can you get convergence of hundreds of genes like that and without some designer doing it? And so we didn't have an answer, but we tried to look for an answer, and that's with the work of Lindsay Kenton in my lab, who's now a postdoc at New England Biolabs. She laser dissected out the song learning nuclei of birds again and the surrounding brain regions and did ATAC-seq profiling. For those that don't know what that is, what we're looking at here is the genomic sequence inside these brain regions like area X and HPC. And we're looking at regions of the genome that are not coded in protein, that are opened and primed for regulation. And the bigger the peaks you see here in front of this gene called EGR1, the more regulation that's region is undergoing to maybe regulate this gene. And for many years, we actually didn't have the regulatory region of this singing activated immediate early gene because it was in a very GC-rich region of the genome that the old sequencing technologies could not get through. With these new genome assemblies, we got it. And what we found for genes that are regulated inside and outside the song nuclei equally, like for singing and hopping, we find that it has peaks of open regulatory genomic regions in front of the promoter, inside and outside the song learning nuclei equally. But other genes like the androgen receptor that is very high in the song learning nucleus compared to the outside, like HPC, we find that there are these open chromatin regions that are high only in the song learning nucleus, but not the outside region. And that's shown here for these two examples. And when we look at that quantitatively, roughly 50% or more of the genes in HPC, RA, and area X that are specialized in those regions have these open chromatin peaks specific to the song learning nucleus and not the surrounding motor circuit. And then we ask those regions that have those open chromatin regions, do they have binding sites for other proteins that would come in and regulate them? That is transcription factors. Is there an enrichment for them? And the answer is yes. For the genes that, like the activity-dependent genes that are regulated equally inside versus outside the song learning nuclei, those genes had binding sites for the transcription factors themselves that regulate immediate early genes, like those singing activated genes. And for the genes that were down-regulated in the song learning nuclei, they were enriched for the NeuroD6 transcription factor. For genes that were upregulated in the song learning nuclei, they were enriched for the androgen receptor transcription factor. And so what's going on here is we think we have a few transcription factors that are regulating several hundred genes to become specialized in the song learning nucleus. These aren't the only transcription factors, but these are the ones that are predominant. And when we take these genes and cross-correlate the ones that have these ATAC-seq peaks for open chromatin regulation and have differential gene expression in song learning nuclei, are they enriched for certain gene function? And the answer is yes. Both the down and upregulated genes are enriched for neural differentiation, axon guidance, as I told you before. But what's interesting, when you look at the human phenotype database and ask for genes that have mutations in them, are they enriched for these genes that are convergently specialized in humans and songbirds? And the answer is yes. And they're enriched for functions that are involved in language impairments, neurological speech impairments, particularly for the upregulated genes in area X here. And so now we want to study, the specific mutations in the genomes, the individual nucleotide changes. And the way we went at that for this gene NeuroD6 is we found its binding site or the open chromatin region in front of NeuroD6. And we found that some of these open chromatin regions had accelerated mutations, these black tick marks here, in all the vocal learning bird groups, shown by these red peaks here. And what this shows, this is a particular part of chromosome 2 of the songbird. And we find there's a peak of rapid evolution here in each of the vocal learning bird species. And when we look at that sequence here in front of the NeuroD6 gene, we find that in the red text here, all vocal learning species have two mutations, one that they share and one that's unique to them, that took this very conserved sequence. We don't know what it's doing in birds in general, but this very conserved sequence and converted into an androgen receptor binding site only in the vocal learning species. Non-vocal learning suboceans actually had two mutations, but it looks like but it did not convert it to an androgen receptor binding site. And so what this would predict is that androgen receptor would bind to this newly mutated, let's say, repressor region only in the vocal learning bird groups to downregulate NeuroD6. When you downregulate NeuroD6, that will then prevent other axon guidance molecules like SLIT1 from turning on, which NeuroD6 regulates, and to shut it off in the vocal learning and speech areas. But how are you going to downregulate NeuroD6 and SLIT1 inside the speech areas, but not, or song areas, but not in the surrounding brain regions? And what we argue is that this newly acquired repressor gets acetylated or a histone surrounding it, preventing the androgen receptor or other repressors from binding in the surrounding motor areas. And so that's the model we're working on going forward. And there's a certain number of genes we think that follow this model that have this convergent expression of open chromatin peaks and with accelerated evolution in these regulatory regions. And some examples here are the androgen receptor itself, ZEB2 transcription factor, NFX1 transcription factor, some axon guidance receptors like plexin-1, SLIT1, and a glutamate receptor, GRIN2B. And so ZEB2, when it's mutated in humans, here's an example of a child with this mutation. Here's what happens. That was trying to say bubble.
Bubble. Yes, put it in.
So like some other deficits, Bella has good auditory or decent auditory learning and perception skills, has difficulty producing learned speech, has other deficits as well, but speech is more particularly affected. ZEB2 has specialized expression, particularly to the dorsal laryngeal cortex. Some genes like RIN2B, we find that it's downregulated during development in the song learning nuclei of the vocal learning birds. Here it's more downregulated in the L-man song nucleus than it is in surrounding brain regions that was in the Nordine's lab. We were able to overexpress the human version of NR2B in L-man and try to bring this bird back to a juvenile state where it's high. We find that the bird song starts to become more variable here. When you overexpress NR2B and L-MAN, an unfortunate side effect, particularly when they're singing directed songs to the females, they start to stutter on a syllable at the end of the vocalization. But we'd like to really try to see what we can do with this direct projection. Could we break the direct projection and prove that it's required for vocal learning and see if these specialized genes are involved? Could we actually get a non-vocal learning species to have this direct projection, take over an existing modal learning circuit and get a quail or a chicken to imitate? And we like to try that with a non-human primate, as the people can imagine what that would be like. But what about a mouse? And at the time we were thinking about doing such experiments, no one really knew what the circuits for mice vocal communication were like, or even if mice, what mice vocalizations were like in terms of vocal learning. We just assumed it was a non-learner. And so the last part of my talk, the last 10 minutes, I will talk about this continuum hypothesis that started with these mice experiments. So it turns out Tim Hawley's group and others showed that mice, particularly male mice, have these ultrasonic vocalizations, sound like this when you pitch it to human hearing range. And this male is trying to produce these vocalizations to court this female. And it sounds like a songbird song when pitched down to our human hearing range, but it turns out to be mostly innate with a little bit of plasticity. When we looked at the forebrain and when these mice vocalized, to our surprise and many other people, there was a cortical and basal ganglia region that showed vocalizing driven gene expression like what we found in songbirds in these mice. And when we looked at the connectivity of these regions, when Gus injected transsynaptic tracers in the larynx muscles, we found that those tracers went from the larynx, went back to this same region that showed the vocalizing driven gene expression, particularly layer 5 neurons, shown at higher power here. And when we inject tracers into this region with these layer 5 neurons, we found that they send axons down to the motor neurons here that control the voice in brown. However, unlike vocal learning birds and humans, there were very few axons that touch these motor neurons. And shown here for birds, there are hundreds of them that synapse onto the motor neurons for vocalizations. In humans, the limited connectivity data out there on postmortem brains indicates that there are hundreds of them as well, these little black squiggly lines surrounding these vocal motor neurons in nucleus ambiguous. And shown again here for the mice, there are only 2 to 3 per motor neuron. So maybe there's a difference of degree here. What about the molecular profiles? Here is this downregulation of SLIT1 gene. We don't see the downregulation here in the mouse motor cortex for the region that projects to the vocal motor organs. What about the physiology? We had somebody, Christina Simonia, said, I won't believe it until you stimulate this region, you get vocal motor fold contractions. And so Zed Vargas in my lab just did that and found that there's 2 areas when he stimulated in the mouse motor cortex here. This is the front of the brain. This is going towards the back, the side, and the middle here. He found an area that I just showed you earlier that was activated with vocalizing activity and gene expression. When he stimulated there, he got both laryngeal muscle and jaw muscle contractions by stimulating that region. And the oral facial cortex region that was supposed to be in mice also found jaw and laryngeal contractions, but stronger for the jaw. And what was interesting, the timing was faster for this oral facial cortex region than here, indicating monosynaptic as opposed to disynaptic projection. However, looking at a 1-pulse stimulation versus a 4-pulse stimulation of that cortical region here, the region where we saw the vocalizing driven gene expression had the most robust activation of the larynx, whereas this other more interior region had more robust activation of the jaw muscles, but not the larynx. You had to stimulate at high levels of stimulation to get the jaw region to activate the larynx. So we think there is segregation here between the larynx and the oral facial musculature in the mouse brain, with greater connectivity to the oral facial musculature than the larynx region connects for the larynx. And so we asked, do these neurons overlap? To some degree, and we injected neural tracer in both the larynx and the jaw muscles for 2 different colors. red for the larynx, green for the jaw muscles. And we found they are some neurons in the same cortical area, but they're mostly segregated, single labeled neurons for each muscle. Although in yellow, we found some double labeled neurons shown in the gray bars here. And so the hypothesis we're working on here is, like Roger Lemon proposed a while ago, is maybe there's a continuum here, and it's not all or none, where like for manual dexterity, the more innovation you get, these gray lines here of axons synapsing onto the motor neurons that control the hand movements from the cortex, then the greater fine motor control you have. Maybe that's what's going on for vocal learning. And instead of inducing a cortical region in the mouse, maybe we can enhance what's already there. And so I'll end off with the experiments we're doing that are trying to move in that direction. And so the hypothesis, why downregulate some of these axon guidance genes? Well, these genes are repulsive in their functions. That is, when plexin binds to its receptor, or SLP binds to its receptor, ROBA1, from, let's say, from the cortex down to the brainstem motor neurons, it repels those axons from making a connection to the neurons that control the muscles. However, in the speech regions, plexin is turned off or turned down, so is SLP1. And we think that then allows these cortical neurons to have direct axons to neurons that control the voice. And so can we mimic that? Here in the human brain, plexin is turned down in the layer 5 neurons. And what we did is created mice that have plexin knocked down specifically with a promoter that's regulated in layer 5, but not the other layers here. And in these mice, what we found, let's look at wild type first. Here is wild type mice. Here is nucleus ambiguous labeled in green. And here are the axons coming from the cortex. You can see the surrounding nucleus ambiguous, very few going through it. And now with the mice with the plexin knockdown, we have these axons now going directly to the motor neurons that control the voice. Here compared wild type to the plexin knockdown. And we can show quantitatively in the homozygous knockouts have more innovation than the heterozygous versus the wild type. And what we find in these mice, they mature their vocalizations faster than wild type. Mice mature vocalizations, they get more sharp in their pitch and their bandwidth acoustic properties. And so these mice move faster during development.
Your name?
And we also show, I'm not showing you here, they produce more complex vocalizations. So for the sake of time, I'll show this result. There is a gene that regulates SLP1. It's called FOXP2, and many of you have heard about FOXP2 before. And we have been studying FOXP2 in songbirds and mice. And when the FOXP2 transcription factor is mutated in humans, it causes the following speech deficit.
Your name?
Laura.
Well, she's trying to say Laura.
Where do you live, Laura?
Sheffield.
And how old are you?
4.
So she's a 4-year-old child that has difficulty producing speech, and like some of the other deficits, has good auditory comprehension and auditory learning. We put Laura's mutation into mice, and we found that in wild mice, they have these two kinds of songs they sing, this more simple one and this more complex one that they sing to females, even though mostly innate. We found that these wild type mice, the mice with the FOXP2 mutations, have difficulty switching to this more complex song. And so, and we find that the layer 5 neurons that project to the vocal motor neurons are still there in the FOXP2 knockout mice, except that they're spread out over the cortex more than they are in the wild type mice, where they're primary localized in the primary motor cortex. And guess what? The females, when they're listening to these songs, they prefer the more complex songs than the simple ones. We call them the sexy songs. So we like to think of this as a continuum, as a continuum here, where it's not an all or none. You have some species that don't vocalize at all. They're simple vocal learners, to the more complex high vocal learners like parrots and humans. And you need these mutations piled on top of each other to cause more advanced and more advanced and more advanced vocal learning to occur, with humans being the more advanced. I'm going to skip the next part just to say here that this one result here, we find that even a continuum within vocal learning species, the more advanced vocal learning skills here you have, the more advanced ability you have to imitate human speech, the greater song repertoire you have, putting all those things together, the better problem solving you are amongst songbirds, and the more innovation that you have amongst the songbirds. So there's a nice correlation with problem solving, innovation, and vocal learning, but not associative learning or other types of learning. So I'll end off with saying we can learn a lot from studying distantly related species about our human speech circuit as convergently involved circuits down to the level of individual cell types and individual connections in songbirds and in humans for a vocal production circuit and a vocal learning circuit here. And I argue that since if several birds and mammals can do this over the last 300 to 65 million years in a similar way with these direct projections to the neurons that control vocal organs, well, maybe a lizard will do it, an amphibian will do it, and I can tell you what their circuit's going to look like and what their genes are going to look like another 500,000 years from now, even I won't be around. And we're getting closer to understanding why can't our closest relatives do what we and parrots can do? And maybe one day we'll be manipulating their genes to get them to say apple. I think that day is upon us. And so end off with 3 summaries here. Complex behaviors can evolve multiple times from deeply homologous but diverse brain circuits and genes. Trait-like spoken language can be understood from studies in distant related animals, and convergent evolution of mechanisms for a trait can be associated with similarities and disorders of those traits. And I gave credit to the people along the way, but there have been a lot of people involved in these projects, and I'll give credit to the funders here and take questions. The ones that are in stars are the ones of the work that I presented today. Thank you very much. And are there any questions in the chat, or you can put questions in the chat or otherwise? I left everybody speechless?
Yeah.
Okay.
I don't think so,
Albert has his hand up, and Linda,
there's already
no,
3.
that's not right. So I guess Albert, you had your hand up.
Okay.
Really great talk, and I'm very curious about the result of the remarkably high correlation between the innovation problem solving ability and the vocal learning ability. Are there common like upregulations of genes that are underlying the projections between the vocal areas to motor areas and like some frontal area projections? Like, is that the reason why that this might be? Like, are there theories about it, or are there experimental evidence about it?
Yeah, so we don't know, but all those birds, we had 15 species in that correlation analysis. Actually, these were wild birds we caught from the wild at the Rockefeller Field Center and then did tests on them in captivity. So this is the real deal. It's not like some kind of laboratory fluke. And so we're sequencing their genomes. We're going to look at the brain expression. We don't know the answer yet, but I bet you there's going to be some differences. We do have a hint, though, for another associated trait with vocal learning species. And it turns out only vocal learners can learn how to dance, synchronize their body movements to rhythmic beats and sounds. And we find in some of those vocal learning species that are the better dancers, they have some of these genes, not all of them, but some of them immediately specialize in the surrounding areas of the vocal learning circuits. And so I think that maybe some of these genes are going to influence other circuits in a way that allows more complex behaviors as well. So stay tuned.
Thank you.
Beautiful talk, Eric. So many cool ideas, and the fruition of other people's ideas, like the different layers lining up, coming true, right. Matching to the circuits. But my question is about the your manipulations to enhance the vocal learning and control of the vocal control with genetics is really exciting. And I wonder do you think if you succeed in connecting up to the 12th cranial nerve through an HVC and RA-like structure, or analog, that the motor pathway, this anterior forebrain pathway through the basal ganglia will just come along for free? Or do you think that there was an additional, genetic changes in that pathway to reproduce a unit? Or is it just the topography of the musculature that's represented there that just gets pulled in?
Yeah, I'm hoping it comes along for free.
Yeah.
Right. That the basic connectivity is there with some small differences. I don't think you need to specialize all those genes to get a forebrain control of the vocal organ. And so I'm hoping it comes along for free and that the specializations occurred later in evolution is my thinking.
And are the genetics more unique in that HVCRA pathway than they are in the basal ganglia loop pathways?
Oh, I would say, I don't know the answer to that. Since L-man has more specializations, I'm thinking probably the other way around.
And area X?
Area X, I mean, area X, no, it's about the similar as HPC in terms of the number of gene specializations.
Okay, it's not garden variety still.
No.
Yeah.
If this doesn't work, then we'll have to figure out how to induce a whole brain pathway duplication. And I have no idea how that's going to work. Maybe it's a gene duplication mechanism.
Thank you.
Guess Martin was next.
Yes, excuse me because I had to walk away for a few minutes if you cover this, but I'm interested if it is still true information that songbirds learn their particular song from their fathers.
Okay.
And what gene expressions or hormonal influences might allow that to occur?
Yeah. So in the Northern Hemisphere, where lots of us study birds, actually the further you get away from the equator, the more often you find of songbird species, females don't imitate, but the males do. But what really seems to have happened in evolution is vocal learning evolved in both sexes first, like in humans, at the origin of songbirds and also parrots and hummingbirds, and then was lost in certain species, particularly when they went to more challenging environments where there was selection for division of labor between the sexes. So what this means, or what we're seeing, is that female songbirds that don't sing, they hatch or born with the presence of song learning nuclei. And as they grow through their critical period, time period, they lose those song learning nuclei. And Mark Kanishi found if you give extra doses of estrogen to these females at a young age, that will prevent the loss of the vocal learning nuclei. And we've looked at the gene specializations when we give extra doses of estrogen, and they recapitulate about 90% of those specialized genes in males. So loss of song in females was selected upon.
Thank you.
Yeah, you're welcome. George?
Yeah, hi Eric. It's nice to see you again. It's been a long time, and since I did any work on this. Actually, you kind of answered my question in a way. I was thinking about female zebra finches that don't have an area X, for example, and they most of them don't have an area X. Some end up with it, but they do have a really prominent L-man. And I was just wondering if, without any hormonal manipulations, have you looked at gene expression in areas like L-man? Because I guess what I'm getting at is that the females need to learn to match onto a song type that they really like, right? So in that way, that is a kind of vocal learning, even though it's not an expression, kind of a bit like Coco, she can express it in different ways. So have you looked at anything like that in the females?
So yeah, this is a good question. We haven't looked at adult females yet for the tiny bits of song nuclei that are remaining. L-man has a little bit more that's remaining. HPC looks entirely shrunk practically, right? So we haven't looked in the adult animals, but it is a question that we are going to address. But what we do find during development so far is as the females lose their song learning nuclei, they lose HPC and RA first. Area X never develops, it never shows up. And L-man is the last to shrink down, even though it never gets down to a really small size. So my prediction is L-man is going to still show some gene expression specializations. And who knows, it could be used for some perceptual processing. I find it hard to believe it goes against all my other hypotheses, but I think you could be right, based upon the recent data. Yeah, and it has been a long time since we saw each other. I wish I realized.
Yeah, so
yes.
I'll let other people ask questions, but it'll be nice to chat soon.
All right. Yes. Nicoletta?
Hi, my name's actually Alexis. I'm in a room full of people watching your talk. Excellent talk. Hi. And my question is, so you seem to support this idea that these circuits for vocal expression in birds are similar to circuits for speech production in humans. I'm curious what you think about human singing and the fact that some people who have impairments in speech production don't seem to show impairments in song production.
Yes, so I was just, Eddie Chang was here at Rockefeller yesterday telling us, and I knew about these kind of findings before, but he puts electrodes into people's brains and records them, as I was telling earlier, with speech production. He's now getting them to sing as well. And the same areas are active in a very similar way, or similar level of activity with singing and speaking. And what he and I, myself and others are thinking is that singing and speech are using the same areas just in different ways. And the explanation as to why some people with Broca's aphasia can sing still more decently than not is that the right side might be a little bit more dominant for singing than the left side is. And so you maintain some of that dominance on the right for your more artistic side, your right side, than your left side. That's the only explanation I know of so far. Nina's here; she might have a better explanation than that. But that's the one I believe in most of things.
Great, thank you.
Yeah. Frederick.
Yeah, thanks for that great talk, Eric. And I'm so impressed that you're trying to basically like doing this enhancement of function, right? Yes. And it's made me think about, okay, it seems like it's relatively simple, not to kind of like undermine this thing. You just have to downregulate a few genes that are involved in repulsion. So there must be a tremendous cost to do this. Yes. Otherwise, this would have evolved like in many, many, many species. And I wonder if you thought about what is the cost of this? Why isn't this like more common? Why don't we have more downregulation of these genes to get this direct control of our cortical to our motor structures and be super good at all kinds of motor skills?
Right, exactly. It's like a trait that seems like why is it absent from so many species if the similarity is already there? So the possible explanation for that came to me, and Kaz Okanoya as well in Japan. But when I was at Duke University, hearing some song sparrows sing from the top of a tree, and he kept singing one song after another, and marching through them, keeping my attention. And I'm thinking, this is dangerous, right? All these predators out here hearing you're at the top of a tree and changing a song all the time. And this what made me thought that predation might be selecting against vocal learning. And if you look at, not all of them, but the number of the vocal learners, they're at top of the food chain. Humans, whales, dolphins, elephants, or bats that sing in the ultrasonic range, so they're not top predators, right? And amongst the birds, that phylogenetic tree we generated showed that songbirds and parrots are descended from apex predators that once roamed the earth, but are no longer here. So I think laboratory mice, since we're not selecting against it, one day might be selected for vocal learning if we breed them long enough. All right. Julie?
Yeah, very nice talk. I was wondering, so when you do this enhancement of function, you are increasing the number of like fibers that are going through that region, right? And do you have a quantification of how much synapses you have actually with nucleus ambiguous? Like, is this really connecting? Is this just passing through, or like, and in terms of activity, then what's going on in the motor cortex that is magic, right? About having
in terms of infectivity?
In terms of neural activity in motor
activity.
Are you trying to look into it?
So we're doing all those experiments now where we're looking at synaptic markers of pre and postsynaptic markers to make sure they're forming functional synapses that, well, at least at the molecular level, they're stimulating the cortex to see if we get more rapid firing of those muscles with shorter latencies. So it's now monosynaptic, as well as putting electrodes there and getting mice to sing. So the graduate students are working on those experiments right now as we speak. So you'll have to invite me next year or at some point to give you those answers.
Okay,
yeah,
all right. Sounds great.
But I'll give you a hypothesis, just to end this off with, is that what I think is going on here in terms of the cortex. I think the cortex has, and HVC and RA, has some type of computational rules to them that controls these motor neurons to produce rapid coordination for auditory motor outputs, sensory motor, input output. And the brainstem doesn't have that ability in these brainstem circuits. And so you need direct control from these layer 5 neurons and whatever is feeding into them to get that rapid coordination. And this is why that happens, and you can't do that with an innate brainstem circuit in the brainstem.
Right, but that's rapid coordination of only the larynx. What about like the other upper vocal tract?
Yeah, right. So it turns out the RA nucleus does innervate muscles, not direct innervations, by the way, indirect to the beak muscles and so forth. In humans, there's also innervation. But in non-human primates, there's also direct innervation for the tongue muscles. So it seems like the larynx muscles is the last one that's missing. You see, the other oral facial innervation from the cortex is there. I showed you for the mice, the jaw muscle has decent innervation.
I see. Okay.
The
Thank
larynx is missing. The syrinx in birds is missing. All right, I guess I finished up here then.
Yeah, I think I don't see any more talks, and I think we could all thank you again, Erich, for giving this presentation, this awesome talk, and for taking your time of your schedule to talk to us. It's been a pleasure. I don't know if anybody else wants to say anything to Erich out loud before we log off, but thanks again.
It was a real tour de force, I think.
All right, thanks.
Eric, I will email you. I'd love to chat with you soon.
Sure, go right ahead and do so. And good to see colleagues again.
Yeah.
Thank you so much.
All right, bye-bye.